Assalamualaikum warahmatullahi taala wabarakatuh. My name is Muhammad Syahiran bin Muhammad Ruslan, who will be presenting this topic, selective DNA targeting through protein-mediated DNA recognition. At the end of this presentation, I'll be discussing on question 1, which is about a non-essential and an essential element for the design of drugs and diagnostic agents. Let's begin with the introduction, where I will first explain what actually selective DNA targeting and protein-mediated DNA recognition are, respectively. Right. Imagine you're a scientist working on a new cancer treatment. You know that cancer cells have different genes and proteins compared to normal cells, so you decide to use this to your advantage. You design a drug that can specifically interact with certain parts of the DNA in cancer cells. This is what we call as selective DNA targeting. The beauty of this strategy is its selectivity. The drug is designed to target only the cancer cells, leaving the normal cells untouched. This reduces the potential for harm to healthy tissues. So, what about protein-mediated DNA recognition? Imagine you're a scientist working on a new type of cancer treatment. You are working with special drugs called metallo drugs, which contain metal atoms. Your challenge is to design these metallo drugs so they can interact with specific sequences or structures of DNA in cancer cells. This is important because cancer cells have different genes and proteins compared to normal cells. Now, designing a drug that can do this is not easy, but you have a strategy. You decide to use what's called a presenter protein. This is a protein that can bind to both the small molecule, which is the metallo drug, and the target, the specific DNA sequence. The presenter protein serves as a bridge, helping the drug reach its target. So you design your metallo drug, and it binds to the presenter protein. This presenter protein in turn binds to specific DNA sequence in the cancer cells. This allows the drug to interact specifically with its target DNA sequence, increasing the effectiveness of the treatment. I have chosen anidine's antibiotics to discuss more on this. Anidine anti-tumor antibiotics are special medicines used to treat cancer. They work in two steps. The first one is activation. These medicines get activated inside the body. Next, once activated, they go to the DNA in cancer cells and cut it. This stops the cancer cells from growing. The special thing about this medicine is the structure. They have a ring shape with some double and triple bonds. This unique structure helps them in the fight against cancer. The first anidine antitumor agent was neocarcinostatin, which was isolated in 1965. Further anidines, principally asparmycins and calicamycins, were not isolated until many years later. So, the first compound of interest under anidine antibiotics is neocarcinostatin. Let's move on to the next slide. This is the structure of neocarcinostatin. Here is the mechanism of activation of neocarcinostatin by thiols. So the mechanism starts with the interaction with DNA. Inside neocarcinostatin, there's an active part called a nephthyoid ester. This part has a special job. It fits itself into the DNA of cancer cells, just like a puzzle piece fitting into a puzzle. Now it also has another part called the DIN. Once the nephthyoid ester is in place, it positions the DIN part within a specific area of the DNA molecule called the minor groove. The second step is activation with the thiol. The compound is activated through a reaction with the thiol. This activation triggers what's called a Bregman rearrangement. Next is the formation of diradical. This rearrangement produces a diradical, which is somewhat unique compared to other anidines. While it's not exactly the same as a 1,4-dihydrobenzene diradical, it behaves similarly. The fourth step is DNA transition. The reactive diradical causes breaks in the DNA strands, disrupting the DNA normal function. But it's not just causing random breaks, it is specifically reacting with certain atoms, which is C4 and C5 of the deoxyribose sugar in the DNA. These specific atoms are usually found at certain points in the DNA sequence, such as deoxyadenosine and thymidine residues. By causing breaks at these specific points, the radical species is effectively disrupting the cancer cell's ability to function properly. The last step is the consumption of oxygen. Each strand break consumes one equivalent of oxygen. This consumption of oxygen is crucial for process of DNA scission. So, to summarize this, neocarcinostatin interacts with DNA and induces strand scission at specific sites. The positioning of the compound within the DNA minor groove suggests a level of specificity that is mediated by proteins. Proteins facilitate the binding of neocarcinostatin to the DNA or modulate the DNA structure to enhance the compound's affinity for its target sites. Moving on to the second compound under Anidine's antibiotics, which is asparagines. Here is the structure of asparagines. And this is the mechanism of activation of asparagines. So let's look at the mechanism. The first step here is the reduction of thiolate. The proposed activation mechanism of asparagines initiates with the reduction of the trisulfide compound to a thiolate. This reduction is facilitated by either an ADPH or thiols present in the cell environment. Following the reduction, an intramolecular Michael addition takes place within the molecule. This addition sets the stage for further chemical transformations. The third step here is the Beckmann rearrangement. The Michael addition here is succeeded by a Beckmann rearrangement. This rearrangement is a crucial step that leads to the formation of the 1,4-dihydrobenzene diradical, which is the activated form of asparagine. Take note on this. So similar to neocarcinostatin, the production of diradicals causes breaks in the DNA strands, which eventually disrupts the cancer cell's ability to function properly. Proteins facilitate the binding of asparagines to the DNA or modulate the DNA structure to enhance the compound's affinity for its target site. Here you can see visually the three-dimensional structure of a complex between asparagine A1 and a DNA duplex, showing the binding of asparagine A1 in the minor groove of double-stranded DNA. In conclusion, the fight against cancer is a complex and multifaceted challenge. However, through strategies like selective DNA targeting through protein-mediated DNA recognition, we are making significant strides in developing more effective treatments. The use of anidines with the unique ability to interact with specific DNA sequences exemplifies this progress. So back to question 1, I've chosen gold as a non-essential element, while zinc as an essential element. Gold is a non-essential element, meaning it's not required for normal physiological function in humans. However, gold compounds have been used for centuries for various medical purposes. One notable example is the use of gold compounds in the treatment of rheumatoid arthritis. Compounds such as sodium aurothiomalate and auranofin have been used as disease-modifying anti-rheumatic drugs, DMARDs, to reduce inflammation and joint damage in patients with rheumatoid arthritis. These gold compounds work by modulating the immune response and reducing the activity of inflammatory cells. In terms of its medical potential, apart from rheumatoid arthritis, research is ongoing to explore the potential medical applications of gold nanoparticles in areas such as cancer therapy, drug delivery, and diagnostic imaging. Gold nanoparticles have unique properties that make them promising candidates for targeted drug delivery and imaging agents in various diseases. Additionally, gold-based drugs continue to be investigated for the potential in treating other autoimmune diseases and conditions involving inflammations. How about zinc? Zinc is an essential trace element required for numerous biological processes in the human body, including immune function, DNA synthesis, wound healing, and protein synthesis. It serves as a cofactor for over 300 enzymes involved in various metabolic pathways. Zinc deficiency can lead to impaired growth and development, immune dysfunction, and increased susceptibility to infections. In terms of zinc medical potential, zinc supplementation is commonly used to prevent and treat zinc deficiency, especially in vulnerable populations such as children, pregnant women, and individuals with certain medical conditions. Additionally, zinc has shown potential therapeutic benefits in various health conditions, including the common cold, diarrhea, age-related macular degeneration, and wound healing. Zinc lozenges or nasal sprays are often used as over-the-counter remedies for reducing the duration and severity of cold symptoms. Furthermore, zinc oxide nanoparticles are being investigated for their antimicrobial properties and potential applications in wound dressing and topical treatments for skin infections. Additionally, zinc supplementation has been explored as a potential adjunctive therapy in the management of COVID-19, although further research is needed to establish its efficacy in this context. Alright, so that's all from me. Thank you.